Hello, everyone. This is going to be one of the few pre-recorded lectures. I usually do them in person, and then we'll get back in person on Thursday, the 18th, for Chapter 8. But let me get started here with Chapter 7. Focuses on the membrane, the cell or plasma membrane structure and function. So this is a continuation of what we covered at the end of Chapter 6, especially the second to the last page of the notes in that Word or PDF doc that I gave you. Starting off with the compounds that form the cell membrane in it, but we're going to go into a lot more detail as far as function goes. So if you recall in chapter 6, we talked about the compounds required to form the cell membrane, and I wanted to focus on two in particular compounds, organic compounds, two of the four, lipids and proteins. And from the lipids I mentioned in chapter 6 that the main lipids were phospholipids. They're the major compounds they're the compounds that primarily form the cell membrane of a cell. But I also mentioned cholesterol contributes to the formation of cells. So we'll briefly talk about that, but we'll focus heavily on these phospholipids. And then there are also proteins, as we talked about before, that contribute to the formation. But starting with phospholipids are referred to as amphipathic molecules because if you recall, they contain both hydrophilic, as it's showing you here, and hydrophobic portions. If you recall, the smaller molecules that form this organic compound are a molecule of an alcohol called glycerol that forms a backbone, and it has three functional hydroxyl groups. Attached to two of those hydroxyl groups, we have two fatty acids. We'll talk about this. Notice how one of the fatty acids, the carbon backbone, is represented by this little, I call it serrated line here, whereas the other one has a serrated line, but notice there's a double line here, and then there's a bend. So we'll talk about this little dotted or serrated line here that they're showing you represents the carbon backbone, and therefore the lines themselves actually represent covalent bonds. We'll talk about this, about where you can get a little bend in the fatty acid, or kink as they call it. That third hydroxyl group, functional group of glycerol, attached to it is a phosphate group, one of our 7 functional groups also, and then attached to that is a hydrophilic or polar head group, which is hydrophilic. I'm sorry, so I meant to say polar head group. There are different types. You take an advanced course, they'll tell you. Phospholipids are given special names based on the polar group. So I'm giving you an example of one known as phosphatidylcholine. I'm not going to test you on that, but just to let you know, there are actually different types of phospholipids. But this choline here, that's where we would rewrite it as just polar group for this class. As we said, we take these phospholipids, and if we're talking about the cell or plasma membrane, these phospholipids form what we call a bilayer. We have layers of phospholipids. One layer we have here, and our other layer we have here. And then in the middle here, you can see between each layer, we have our fatty acids, which are facing each other. Why? Because inside and outside of the cell, what we primarily have is a water-based solution, or what we call an aqueous solution. Solids dissolved in the solvent water. Okay. So when you look at these two phospholipid layers, the overall part here is a phospholipid bilayer, okay, consisting of two layers of phospholipids. Now, where you have these fatty acids meeting, where they are meeting right here, they create a barrier. Since fatty acids are actually hydrophobic, or what they sometimes refer to as hydrophobic tails, what we end up with here is a hydrophobic barrier. Okay, so if you look at the figure over here that they're showing you, taking this and using the other diagram or representations of the model that I gave you on the previous figures, notice here they're showing you the polar water molecules on each side of the bilayer or membrane. But notice the phospholipids, this part right here where you have the fatty acids, notice they don't show any water molecules right here. Now in some classes that you take, even our A&P classes, they don't like to go into too much detail about how water can move across the cell membrane of our cells, with rare exception, and the only exception I know of is in the kidney. So in this class, we'll talk about how the membrane of our cells and cells in general are permeable to water. But we'll talk about in this class, it's because there are water channels, okay, transmembrane proteins that function as water channels. You'll come to know as aquaporins, and I'll mention these again in a bit. Now, that phospholipid bilayer, as I said before, it forms a barrier, okay? But it's not an impermeable barrier. The barrier is what we refer to as being semi-permeable. Sometimes they will say selectively permeable. Okay? Often in advanced classes, They say selectively permeable. I say advanced because I used to teach a senior level cell and molecular course. I don't remember half the stuff I taught, but it definitely was advanced compared to this. But they'll say selectively because, as I'll talk about as we go through this chapter, it's not just about the phospholipid bilayer formed by phospholipids, but it's also about proteins, specifically transmembrane proteins. It's a combination of the two that contribute to our cell membrane being semi-permeable. However, when we get into those transmembrane proteins have many different functions, but the ones we're going to focus on in this chapter, okay, play a role in membrane transport. And these transmembrane proteins, they create little pores or doorways in the cell membrane, but they selectively allow certain compounds or ions to pass through. They don't just allow anything. Okay, it's not a doorway where anybody can pass through. It's a doorway with a checkpoint, and you got to have the right ID to get through. So this is what we're talking about here. Our cell membrane is semi-permeable, okay? But it just meaning that some molecules and ions can pass through, others can't. This is the best figure I have right now, but I don't like this because they should have shown lipids going through instead of water and ions. So it's a little backwards, but the purpose is just to show you what we mean by semi-permeable or selectively permeable. Okay, so if you look here at the cell membrane, they're showing you here, we have our phospholipid bilayer. Okay, they're not showing you cholesterol. I'll show you a figure on the next slide that has cholesterol, but they're showing you the phospholipid bilayer that's making up the majority of this cell or plasma membrane, as they show you right here. Okay, I'm going to start saying cell membrane primarily from here on out. I'll mention plasma when we one more time or so, but know that it is. Please do know that it's known as a plasma membrane. I know four-year colleges primarily use this term. But if you look at this cell membrane, okay, the primary compound are phospholipids. They form that bilayer. But then they show you these guys here. Okay. Those are transmembrane proteins. Okay, and if you notice, they're showing you these transmembrane proteins, but they're trying to show you over here and here that they create a little doorway or pore. Okay. Some substances, including ions, okay? So you're going to hear me say substances a lot because it's not just molecules, it's ions, and ions are not molecules. So some substances, okay, a cell needs to cross the membrane, either because the cell needs to have, move from the ECF to the ICF, or the cell needs to get rid of something, therefore it needs to be transported from the ICF to the ECF. Now keep in mind, we say ICF and ECF. ECF is up here, stands for extracellular fluid. Inside of the cell, we have the cytoplasm. That part of the cytoplasm, okay, inside of that part of the cytoplasm that is just the solution, it is known as the cytosol or simply the ICF. So we are building off of Chapter 6, and I already went over these two terms here. Okay, so this is the one time I'm going to mention it. Okay, these are both solutions, so you're also responsible for knowing that. Make sure you go back over what a solution is. So we have ECF and we have ICF, extracellular fluid, intracellular fluid. And what happens is cells need certain substances to be able to cross the membrane into and out of the cell. In other words, an exchange between the ECF and the ICF. However, some of these substances are not lipid soluble. They cannot move through this hydrophobic barrier, but the cell needs them to get across. Therefore, this is where your transmembrane proteins are going to come in. So I'm going to erase all this here. Okay, and then we'll get to this point. So if you look here, what they're showing you on this figure, they're showing you up here certain lipids. This right here, if you recall, I said whenever you see these little 4 rings, automatically tell yourself it's a what? Steroids. Steroids such as estrogen. Okay, or lipid-soluble molecules. Well, lipid-soluble molecules and soluble gases, well, to be honest with you, you can just think of those lipid-soluble molecules as being gases, O2 and CO2. Certain lipids, such as estrogen, as well as the gases O2 and CO2, can readily pass through the membrane. In other words, what we call readily diffuse across the membrane. They can actually get across or pass through this hydrophobic barrier. Well, lipids and lipids, it's like a drop of vegetable oil into a cup of vegetable oil. It all fuses and blends together. You can't tell where the drop went, like and like. But why is it O2 and CO2 can move and readily or easily pass through this hydrophobic barrier? They're small, nonpolar molecules. and the cell needs them to be able to move as easily as possible, so it works out beautifully. But they're small nonpolar molecules. They easily move it across the membrane here. O2 will move into the cell, CO2, the waste product, will move out. So these are examples, okay, demonstrating how the hydrophobic barrier here, it's not impermeable. As I showed you on the previous slide, it is semi-permeable. However, if you look over here, water, small water-soluble molecules and ions, they diffuse through the membrane, but they have to diffuse through channels. Well, these are called channel proteins. So where it says channels here, that's what they're showing you here. They're showing you channel proteins. Okay, and if you follow this line here, they're saying here's an example of a channel protein. These little bitty guys here are representing our water molecules. This is representing our small water-soluble molecules. And you can see here, diffuse through a membrane channels that vary in shape, size, and specificity. Well, basically what they're saying is these channels right here, you're going to come to know them as channel proteins. Okay. These are transmembrane proteins that help water and water-soluble molecules and ions, okay, diffuse or pass through the membrane. We'll talk about how diffuse, if I can find it, my eyes are real bad, but we'll talk about right here how when we say diffuse, we're going to talk about this process. It's short for diffusion, a passive form of transport. But we'll also talk about active forms, and I'll explain those later. But the bottom line is you have a channel protein here, and it will allow for certain substances, water-soluble molecules as well as ions, to bypass the hydrophobic barrier and get across the membrane, be transported across the membrane by going through these channel proteins. However, if you look down here, okay, on this part, I'm going to erase all this stuff. But if you look down here, notice something, what they're showing you. Here's a channel protein right here. Oops. Let me do this again. That was a bad direction to draw the arrow. If you look here, notice this arrow here, but notice something. We're still talking about a channel protein right here. Okay. Notice something, however. Down here it says some large molecules cannot fit through the membrane channels and cannot diffuse through the lipid bilayer. They can cross the plasma membrane only by means of a carrier mechanism. Okay, so here this represents what you'll come to think of as a molecule of glucose. Okay. We'll talk about a second class of proteins. Okay. These proteins carry certain substances across the cell membrane, and I'm screwing it up again, carry certain substances across the cell membrane. So you'll come to know there are not only proteins called channel proteins that help certain substances, including ions, bypass the hydrophobic barrier and move across the cell membrane, but there are also other transmembrane proteins called carrier proteins that help move larger molecules, okay, across the cell membrane, whereas a channel protein just can't accommodate them. So what I primarily use this figure for here is to show you, okay, what we mean by calling a membrane semi-permeable when you compare these characteristics here with these characteristics here. But even when you look at these characteristics here for what it takes across a membrane, now you got to look at these characteristics, and it's just basically saying there's not one type, a transmembrane protein, that allows any water-soluble substance or ion to move across the membrane. There are many different types of transmembrane proteins, and for those that function in membrane transport, some are channel proteins and some are carrier proteins. And what they are responsible for transporting across the membrane, okay, that's where we start getting these differences between channel and carrier proteins. Some molecules are just too big to move, but the cell needs them to move. So if you look at this next figure, okay, again, I'm basically on page 2 primarily right now. So page 1 to page 2. So we have our phospholipid bilayer, but it's in this figure here where they show you cholesterol. Okay. Cholesterol, one way I put it in your notes, I believe it's roughly contributes to how fluid the membrane is. Okay. You can simplify that by saying cholesterol contributes to how rigid or stiff the cell membrane will be. Not all membranes of various cells have the same concentration or number of cholesterol molecules in them. It depends on the functions of these. Okay. But you can see here the cholesterol Okay, is that not in, it's not one molecule of cholesterol that's inserted into the membrane that crosses the entire membrane. You'll see molecules of cholesterol in one layer or the other phospholipid bilayer, but you can see throughout here. And then this figure is just showing you different types of transmembrane proteins. Please remember, in the previous chapter, I believe I showed you the figure, okay? Some transmembrane proteins function as CAMs. They will bind the membrane of a cell to the extracellular matrix. Some transmembrane proteins function as CAMs, binding the cell membrane of one cell to another. But I also mentioned how some transmembrane proteins function as receptors, all right? And then some transmembrane proteins, as we're going to focus on, and I'll jump ahead because I forgot I have it, some transmembrane proteins actually function in membrane transport. So that's what this figure is. This is slide 8. It's just to show you a representation of the different functions transmembranes can, play a role in, or how they function differently. Some function as CAMs, binding to the ECM or binding to another CAM in another cell membrane. Some function as receptors that we'll talk about in chapter 11, and then some function in what we call membrane transport. Okay. This figure right here shows you all three types of membrane proteins. Transmembrane proteins are sometimes called integral proteins. Okay. You take an advanced course, that's the only time I've seen this term. When you're talking about an introductory or freshman level course like 1406, okay, you can use the term transmembrane, which means through. Notice they made the phospholipid bilayer real small because the star of the show in this figure is proteins. But if you look here, these are peripheral proteins. You can find them within the cell or outside of the cell, and they're intracellular or extracellular. And then lipid anchored proteins, okay? Here's a protein that is a lipid anchored protein. Here's another one. For lipid anchored proteins, they have a single fatty acid attached to them, and then that fatty acid is inserted into the cell membrane. The reason why you find some of these proteins is because it actually allows the protein itself to move around while staying anchored to the membrane much more freely or easier than these guys are. If you notice some of these transmembrane proteins, they're anchored to peripheral proteins, which are anchored either to the cytoskeleton or possibly to the extracellular matrix. That's not 100% true. Obviously, we're going to talk about integral proteins, which are involved in membrane transport in chapter 11, receptors. So not all of them will be in that arrangement, but this is just mainly to show you the three different types of proteins. And again, keep in mind, integral is a fancy way of saying transmembrane proteins. I will use transmembrane, or I will use a more specific name such as CAMs, receptors, or for this chapter 7, you will hear me say channel proteins or carrier proteins. This figure right here, which I'm going to circle, this right here represents a channel protein, but I'll show you the carriers. Okay. When we talk about the cell membrane, there's a model called the fluid mosaic model. Okay. The cell membrane of our cells, one of the reasons for calling it plasma, or the reason for calling it plasma membrane, if you want to say, is if you think about plasma, normally when you hear about plasma, you hear about blood plasma. Okay, well, blood plasma, it makes up 55% of your blood, roughly 52% or more is water of that 55%. So we're talking something of a viscous fluid or thick fluid. Well, our cell membrane here, as rigid and stiff as it looks in these pictures, is actually very fluid. Hopefully y'all watch the videos that I provided, such as the life of a cell. I hope y'all are watching these videos, but in the video for the life of the cell, and I don't recall if I put it on chapter 7, if I did not add it. Okay, what they try to show you is that the cell membrane of our cells, you got to think of it as looking out on a body of water, whether you're looking at the outer surface or the inner surface. You got to think of it as a body of water, okay? Some parts, such as some transmembrane proteins, such as CAMs that are attached to something outside or inside of the cell, they're pretty much held in place. Okay, they don't have the freedom to move. But what they're not showing you is proteins that are not really anchored to anything, and I don't have a good one on this figure, or maybe this one here. Proteins that are not really transmembrane proteins that are not really anchored to any other molecules or larger structures inside or outside of the cell. They actually float around in the membrane. They will float around in what they call a lipid raft. So these phospholipids here in particular, you got to think of them as being comparable to molecules of water. As stagnant as they may look, there's not. There's movement. They're fluid. Some areas, there's not as much movement because of what the proteins are associated with. Okay, but in some areas like this protein right here, which doesn't look like it's attached to anything on either side, this protein here could conceivably float around in the membrane, moving around as a lipid raft, meaning some phospholipids stay around it. But you never get a break in the membrane. Everything's moving together. So it's like looking out on a body of water. It might look stagnant, okay, but especially if there's a boat or something floatable in the water, that's when you notice, okay, there is some movement even though it looks stagnant. It's not. That's the way. This is the main reason why we call this cell membrane the plasma membrane, and this is where the fluid of this fluid mosaic model phrase comes from. The mosaic part of this phrase has to do with the oops, I guess I took it out. Has to do with the mosaic of different types of proteins. Okay, so going back to that previous figure, has to do with all these different types of proteins associated with the cell membrane, transmembrane proteins, peripheral proteins, lipid anchor proteins. You have a mosaic of different proteins, and you can see as they're trying to show you here, you have many of them. Okay, it's like going out again to a body of water on a holiday like 4th of July. Everybody goes out, goes to the lake. You look out on the lake and you can see the water, and there are a lot of people out there. You can imagine these proteins are the people out there. I know it sounds corny, but I'm just trying to come up with an everyday analogy there. Now, the phospholipids themselves, specifically the fatty acids, okay, because I talked about the fluid part is mainly attributed to the phospholipids. They actually move around. The mosaic part is attributed to the variety of different proteins you have in the membrane, some of which are flowing it around in that fluid part, or the phospholipids. But the phospholipids themselves, okay, are sometimes described as being creating a very fluid plasma membrane, or sometimes they create what we call a very viscous plasma membrane. Okay, which the phospholipids form in a particular for a particular cell's membrane has a lot to do with whether or not, okay, the fatty acids that make up the phospholipids, or part of the phospholipids, are unsaturated or saturated. The way it works, okay, is that if you see these yellow lines represent the fatty acids of each phospholipid. If the fatty acid has a little kink or bend in it, then that means that a pair of the carbon atoms that form the carbon backbone, you have at least one pair of carbon atoms that have formed a double covalent bond between each other instead of a single covalent bond. And whenever two of the many atoms of carbon that form the backbone of a fatty acid form a double covalent bond between each other, that one double covalent bond between those two atoms of carbon creates a little bend in the fatty acid, or kink. However, whenever the fatty acids, okay, or whenever the carbon atoms that form the backbone of a fatty acid are all covalently bonded to each other through single covalent bonds, you end up with fatty acids that have very straight, very linear, or what they call saturated. Now, what's the big deal about this? Well, when they have a double covalent bond, that makes for a more fluid membrane, but it also results in a lot more gaps in the phospholipid bilayer. Well, that's not a problem, because if you look down here, cholesterol will fill those gaps. As opposed from when our cell membrane is described as viscous, okay, or thicker and not as fluid, well, that's when you have what we call saturated fatty acids, or saturated hydrocarbons. In other words, the carbon backbones of these fatty acids, they're all covalently bonded to each other through single covalent bonds. You don't see any doubles, therefore they're tightly packed together. Okay, less movement. Now, what's the purpose of torturing you with all this information? Well, first of all, not torture, but just in case you think of it that way. What we're doing now, so up to this point, but not including membrane transport, what we're doing now is basically we're building on top of the information I introduced to you in Chapter 6 on the cell membrane. Okay, I gave you a little bit of information in Chapter 6. Hopefully you studied that information in Chapter 6 on the cell membrane, so that when we start Chapter 7, hopefully you were noticing, okay, yeah, I know the compounds that he wants us to know that form the cell membrane, but now he's adding on to this. He's giving me a lot more detail. Well, why am I giving you a lot more detail? Because the purpose of Chapter 7 is membrane transport, okay? Now, when I say membrane transport, the way I define membrane transport is this is a transport of substances, molecules and ions, across the cell membrane into and out of the cell. This results in an exchange between the ICF and the ECF. This exchange of substances between the ECF and ICF can either occur through the hydrophobic barrier created by the phospholipid bilayer or through transmembrane proteins called channel proteins or carrier proteins. Now, membrane transport is described as being either a passive process or an active process. All right. Now, to go back a little, I already went over this figure. This is just the same figure I went over at the beginning, reminding you. So it's just showing you, hey, look, here's the figure again. Here's the slide. I'm showing it to you a second time, so it must be important. So make sure you work your way through this, preferably clockwise. Start up here and just work your way around. Okay. Now, I said membrane transport can be a passive process or an active process. Okay. The key difference between passive processes of membrane transport and active processes of membrane transport, first and foremost, passive processes of membrane transport do not require energy in the form of ATP to transport substances across the cell membrane. The key difference with the active process is that active processes of membrane transport do require energy in the form of ATP, and active processes of membrane transport always require transmembrane proteins called carrier proteins. This is what they're showing you here. Now, the reason why active processes always require ATP is because it's the carrier protein that's using that energy. This carrier protein cannot perform a type of membrane transport called an active process, or you'll come to know it as active transport, cannot do its job unless it has the energy to do it. All right. Now, for passive processes, this is what we're going to focus on the most for this chapter. We'll visit active at the very end, but the majority is on passive. Okay. If you look here for passive processes, in addition to not requiring energy in the form of ATP, okay, passive processes may or may not require transmembrane proteins, okay. If the passive process does require transmembrane proteins, the transmembrane protein can be a carrier protein, which is here, or it can be a channel protein, which is here, okay. But they're showing you over here, it does not mean you have to have, okay, a transmembrane protein, right? So again, passive processes never require energy in the form of ATP, and passive processes may or may not require transmembrane proteins called carrier and channel proteins. However, the question then is this: what is the driving process then, okay, that causes passive processes of membrane transport? Well, it's called a concentration gradient. Check my spelling, because right now I'm trying so hard to focus on just trying to write something decent. Okay. Remember, in chemistry, we use brackets to represent concentration. You see me do that a lot in lecture where I can and in lab. That's what the brackets represent. Okay, that's concentration. And the concentration gradient. Okay, we define a concentration gradient, okay, as being an area within one or two solutions. In this case, it's two solutions. Imagine this is the ECF and this is ICF, okay? In this example, we have the way we identify a concentration gradient, okay, is we look at the solution and figure out which of the two solutions has a higher concentration of solute. So I'm going to draw an arrow, and which of the two solutions has a lower concentration of solutes. Okay, if there's two solutions separated by a membrane, and we see in one solution there's a higher concentration of solids than the other solution, then we have a concentration gradient. So again, the driving force for passive processes of membrane transport is that there is a concentration gradient. There has to be an area of high concentration of solids in a solution near an area of low concentration of solids in a solution. Okay, so the term given for a passive process of membrane transport, it's called diffusion. Okay, now diffusion is defined as the movement, okay, of molecules and ions from a high concentration to a low concentration, okay, until equilibrium. That's the definition for diffusion. The problem is every time they show you diffusion in a textbook, all they're showing you, okay, is the movement of solids and never water. So listen carefully. For this class, I'm not 100% truthful, but I do this to cut down on the mistakes. So in this class, if I ask you for the definition of diffusion, this is a definition I want you to know here on slide 14. If I ask you to define diffusion, such as on the homework, you would write down focuses on the movement of solids from a high concentration to a low concentration of solids until evenly distributed, or you can simply put until equilibrium. Okay. Now, this process can occur with or without a membrane present. So when we talk about diffusion, we don't necessarily need a membrane. So if you look here, they have the sugar cube, and they colored the sugar cube purple so we can track the movement of the molecules of sugar that form this cube. Okay. They drop the sugar cube in here into our solvent water. All right. Once it's dropped in here, now you can see where the sugar cube has landed at the bottom of the beaker. This is the area we have of high concentration, and as we move away from the sugar cube, I'm making it worse, as we move away from the sugar cube, notice the area of low concentration up here. Okay, let me do that again. Let me get rid of these arrows. So again, once we drop that sugar cube at the bottom of the beaker here, we have an area of high concentration of solute, and then you can see everywhere else it's just water. So now if you notice over here the little halo, it's showing you how the sugar molecules are diffusing from a high concentration in the direction of a low or no concentration, if you want to call it. But I like to simplify it. You can see here the sugar molecules are diffusing from an area of high concentration to an area of low concentration. And that's what they're showing you here. Okay. Now, as I said, the sugar molecules are stained, or the sugar cube is stained purple. Therefore, the sugar molecules are purple, so we can track their movement. Now, as these molecules of sugar diffuse from a high concentration to a low concentration, eventually we reach equilibrium, which is represented here. So I circled both of them. I guess if I move real fast and draw long lines, I do a better job, huh? Nah, it's just less worse than the others. All right, so if you look here, we're at equilibrium. How do we know in this figure that we're at equilibrium? Well, now we have a sugar solution, but because the sugar molecules are stained purple, we can see the color purple is evenly distributed throughout the water or throughout the sugar solution with the solvent water. So it's just a visual way of representing this experiment. The catch is we're talking about membrane transport. Movement of solids, or substances, I should say, across the cell membrane. So here now let's talk about diffusion, but in the presence of a membrane. If we're talking about diffusion of solids across a semi-permeable membrane, all right, then there are two questions we have to ask ourselves. First, do we have a concentration gradient? Okay, well, in order to determine that, we first have to recognize we don't have one solution, okay, we have two. One here and one here. The reason why is because of the membrane here. It's dividing this solution into two solutions. So we want to ask ourselves two questions. The first question is, do we have a concentration gradient? Well, that's pretty easy. You can see here we have a high concentration of solids on this side. Okay. On the other side of the membrane where we have our second solution or our other solution, we can see there's really no solute concentration. So the first question we want to ask ourselves, when we know a membrane is present, meaning we have two solutions side by side separated by the membrane, we then want to ask ourselves, do we have a concentration gradient? In this example, yes. But the second question is this. We have a concentration gradient, but are the pores in the membrane, the openings in the membrane, are they large enough? Are these pores large enough to allow these molecules of solute to diffuse from a high concentration to a low concentration until equilibrium? Well, in this picture, in this representation, yeah, it is. So you can see here they're showing you. Both questions, the answer is yes. We have a concentration gradient. Yes, the membrane pores are large enough to allow the solute to pass through, or another way to say it, yes, the membrane is permeable to the solute. So since the membrane is permeable to the solute, we have a concentration gradient. Notice the molecules of solutes are diffusing from a high concentration to a low. But then notice this, they use the term net. Net. They don't say diffusion, they say net diffusion. Why is that? Notice, most of the arrows are pointing from our left to our right. The molecules of solute or dye are diffusing from a high to a low concentration. But they're showing you some of them are deflecting off, and then when we come over here, when we go from here to here, notice they're showing this one is not deflecting off the membrane like over here. They're showing you this one is moving in the opposite direction, that the majority of the molecules of solute that are diffusing. Therefore, we say net diffusion. What that means is that the majority of the molecules of solute are moving from our left across the membrane to our right, diffusing from a high concentration to a low, but you do have a few molecules that are moving in the opposite direction. But the majority are moving from our left to our right, so we say net diffusion. I'm saying this over and over again because Lab 5, you're going to have a lot of questions, and you're going to have to read the questions carefully, because the questions at one point will ask you, do you have movement of the solute? Or is there movement of the solute? And then the very next question after it will ask you, is there a net movement of the solute? Movement of the solute just means, can it move across the membrane in either or both directions? Net movement means, are most of the molecules of solute moving in one direction? And then notice here, eventually we reach equilibrium. Okay, now I haven't set it up to now, but what do we mean by equilibrium? Well, the concentration of solutes in both solutions is equal. Okay, another way to say that is, let's say for every 1 molecule of solute, we have 50 molecules of water on this side of the membrane. And over here, if we were to count up the number of solutes and compare them to the number of water molecules, let's say we also come up with the same ratio. For every 1 molecule of solute, we have 50 molecules of water. So since we have the same ratios, we can say we have the same concentration, therefore we are at equilibrium. Now, is movement still occurring of the solutes at equilibrium? Yeah. But for every 1 molecule that moves from our right to our left, one is moving from our left to our right. So there's always movement if the membrane is permeable to the solute. It's just a question, is there net movement? Are more of the molecules moving in one, or substance moving in one direction than the other? Now, why do I go into that detail? It's for this next part. Osmosis. Now, osmosis, first of all, is a special type of diffusion whereby we only focus on the movement of water. The reason for this, okay, please listen carefully. The reason for this is because, let me go back here. The reason for this is because almost every cell of our body has channel proteins that function as water channel proteins. Now, this is the lay term for these channel proteins. In your notes, I give you the technical term. There are proteins that are transmembrane proteins that function in the transport of water. The lay term for these proteins is water channel proteins. The technical term or name given to these water channel proteins, aquaporins. AQUAPORINS, aquaporins. Okay? 99.9999% of the cells of our body have aquaporins. And these water channel proteins are always opened, allowing for the movement of water, the free movement of water. Okay. Now, that's the first part I need you to know. Okay. So going into the test, just simply think of it as cells of our body. We're going to focus on us. Okay. Cells of our body all contain in their membranes aquaporins, water channel proteins that allow for the movement of water or the diffusion of water into and out of the cell freely. Okay. But instead of saying diffusion all the time, we say osmosis. So osmosis is a special type of diffusion, and this is the definition I want you to know. Some textbooks will say osmosis is the movement of water from a lot of water to a little bit of water. If you look up various IV solutions, okay, there are different types of IV solutions. Here in Texas, probably the most popular IV solutions used on a lot of patients are the ones for people who are dehydrated. So look up these IV solutions and you'll notice this is the better definition to know. And I actually got this from an emergency medical care textbook, okay? The definition I want you to know for osmosis is that it focuses on the movement of water across a semi-permeable membrane in the direction of a higher concentration of solids until equilibrium, okay? So notice for this definition, unlike diffusion, we only talk about osmosis occurring in the presence of a membrane. It always focuses on the movement of water. What's the driving force? What's causing water to want to move? Well, water wants to move towards a higher concentration of solids. Now, will water move forever? No. It'll move until the two solutions on each side of the semi-permeable membrane are at equilibrium. And this is a figure they're showing you up here. So if you look here, they have a pipe here. Here's our semi-permeable membrane. Let me do this again. Here we have our pipe here. And they're showing you, here's our semi-permeable membrane. You can see it has small little pores in it. Okay. This membrane is separating two solutions, but both solutions are sugar solutions. All right. The solvent obviously is water. The solid is sugar molecules. All right. You can look here just visually. You can see there are many more green beads on this side of the membrane than on this side, meaning that the concentration of sugar on this side is much higher than the concentration of sugar on this side. So let's take a step back. Focusing on the movement of the solid sugar, does diffusion want to occur? Yeah. There's a higher concentration of solid here than there is here. So the sugar wants to diffuse from a high concentration across the membrane to a low. Remember, I said when the membrane is present, you have to ask yourself the second question. Is the membrane permeable to the solid? Well, look at this figure here where they draw the box and then they magnify the box. Notice these green molecules of sugar and notice the pore size. The sugar molecules are too big to move across the membrane. So yes, we have a concentration gradient here. Yes, the molecules of sugar want to diffuse from a high concentration of solid to a low, but no, they cannot diffuse. They're being blocked by the membrane. The membrane does not contain pore sizes large enough to allow the sugar to diffuse from a high to a low. However, we want to treat the membranes of cells in this class as if they're always permeable to water, okay? Just like in our body, the membrane of our cells is always permeable to water because they contain aquaporins in their membrane, all right? So if you look at this example, what they end up doing is they end up making sure that these little water molecules are small enough to pass through the pore size, through the pores in the membrane. So what happens? The solid wants to diffuse from a high to low concentration, but the membrane's blocking it. However, if you notice, read the definition for osmosis. Does this special type of diffusion where water focus on the movement of water occurs, does it occur? Well, yeah. If you read the definition, osmosis focuses on the movement of water across a semi or selectively permeable membrane in the direction of the higher concentration of solids. Well, we know we have a higher concentration of solid over here, which is represented here. So notice the red arrows here are for the water molecules. Notice there are no arrows coming off the sugar molecules because they can't get across the membrane. So when we focus on this special type of diffusion called osmosis, the molecules of water want to move across the membrane in the direction of the higher concentration of solutes, and that's what they're showing you. Notice most of the red arrows are pointing from your left to your right. Now, but technically we would say, is there a net movement of water? Because you can see here some water is going in the opposite direction. However, there is a net movement of water across the semi-permeable membrane in the direction of the higher concentration of solutes. So eventually what happens is we go from here to here. Notice up here, it says the two solutions have the same concentration of solute. Okay? But if you look here, most students, what they focus on is, no, this has a greater volume or a greater total volume than this one. So they can't be at the same concentration. Concentration doesn't have to do with total volume, everybody. Unless you're making a solution, then you need a total volume. When it comes to comparing two solutions and asking yourself, when it comes to two solutions and asking yourself, are they at the same concentration? It has nothing to do with total volume. Again, in Lab 2, it talked about different ways of explaining concentration of solutions. And it says, and I believe it's on the first page in Lab 2, you can use a ratio. So I'm going to use it again. Even though the total volume of these two solutions is not the same, they are at the same concentration. Hypothetically speaking, let's say for every 1 molecule of sugar I count over here, I have 100 molecules of water. And let's say I come over here on this side, once at equilibrium, and I count the same ratio. For every 1 molecule of sugar I have on this side, I have 100 molecules of water. So even though the volumes are not the same, the ratio of sugar, 1 to water molecules, 100, 1 to 100 ratio, is the same on both sides. Therefore, they are at equilibrium. They do have the same concentration. Okay? Don't be misled by the total volume. It's about concentration. These are at equilibrium. They have the same ratio. Now, when we talk about osmosis and the movement of water, let me go back here. When we talk about osmosis and the movement of water, in a living organism such as ourselves, as I said before, okay, just because we have a concentration gradient for the solute doesn't mean the solute can diffuse across the membrane, because the membrane may be impermeable to that particular solute. Whereas I said to assume the membrane is always permeable to water. Well, this is because that's indicative of what happens with the cells of our body. Down here, they're showing you red blood cells. Okay. When I was a student, we would come to physiology lab, and they didn't give us these labs ahead of time. They gave them to us as we walked in usually. When we walked in, there was a day where we would do a study on osmosis and tonicity. Okay. The best way I can define tonicity, okay, this is the way I define tonicity. Tonicity has to do with how the movement of water across a cell membrane creates stress on the cell. This stress can result in a cell losing its normal shape, as they're showing you here. Okay? Now, the stress created by water moving across the cell membrane, it's described as a type of pulling pressure, better known as osmotic pressure. Okay? In other words, the water's moving in the direction of the higher concentration of solutes. So sometimes they say, hey, think of it as the molecules of solute are pulling the water across the membrane of the cell towards the higher concentration. But the water moving across the cell membrane is creating a type of pressure known as osmotic pressure. This could result in the cell losing its shape. So when I was a student, we would come into the lab, and one student would have to prick their finger and put a few drops into 3 test tubes that were given to us. One of the test tubes would have a salt solution, and I forgot the percentage. I don't remember the percentages, but one of the test tubes would have a salt solution that the red blood cells in the drop of blood would like. They were happy with the concentration of solutes in the salt solution. Why? Because the concentration of solutes in the salt solution, okay, was at equilibrium with the concentration of solutes within the cell. Therefore, okay, solutes. Not all solutes can easily pass across the cell membrane. However, I said water can move across our cell membranes because of those aquaporins. Well, what happens is that water is moving across the cell membrane, but just as much water is moving into the cell as is moving out. Therefore, the cell is able to maintain its normal shape. Well, what it was that we had placed a drop of blood into a test tube, okay, that had a concentration of salt or concentration of solutes that was equal to the concentration of solutes within the cell. Therefore, the solution surrounding the cell, the ECF surrounding the cell, was referred to as an isotonic solution. Okay, so the term isotonic solution is the term given to the ECF. Whenever the solute concentration of the ECF is at equilibrium with the solid concentration of the ICF. This is what our cells want. They want to be in an isotonic solution. However, in another test tube they would give us just had water in it, nothing else, just water. Okay? So when we put a drop of blood in there, the red blood cells did not like that environment. Because why? Because the test tube only had water in it, that meant the higher solute concentration was within the cell. And because not all solutes can easily diffuse across the cell membrane of our cells, what ended up happening was that the ICF had a higher solute concentration than the ECF. Therefore, in this case, what would happen is that the water molecules in the test tube, in the ECF, okay, would move across the membrane into the ICF where there was a higher concentration of solute. There was such an extreme difference in solute concentration that you can see down here, instead of having normally shaped red blood cells described as a biconcave shape or donut shape, they would swell. So up here, we would say the ECF in this case, in this second example, we would say the ECF is behaving as a hypotonic solution. Whenever cells are surrounded by a hypotonic solution, the concentration of solutes in the ECF is lower than the concentration of solutes in the ICF. Therefore, water moves across the membrane towards the higher concentration of solutes and will continue to have a net movement into the cell until equilibrium is reached between the ICF and the ECF. Therefore, in this case, if the ECF solute concentration is low enough, it's not only behaving as a hypotonic solution, but it causes a cell to swell, maybe even lyse or burst. Now, what we're most worried about living here in Texas is this third condition. Okay, so what would happen is we would have a third test tube when I was a student, and we would put a drop of blood into that third test tube that had a very high salt concentration. Okay, that salt solution or saline solution in the test tube that had a very high salt concentration, that was our ECF. When we put a drop of blood into that tube, our red blood cells again were very unhappy. Why? Because our red blood cells now were placed into a test tube where the concentration of solutes in the ECF, okay, was much higher than the concentration of solutes in the ICF. Okay, remember, we're under osmosis. That's what I'm explaining here. So what happens? By placing red blood cells in a solution with a very high salt concentration, the ECF is said to behave as a hypertonic solution. Whenever the ECF behaves as a hypertonic solution, that means the ECF has a higher solute concentration than the ICF's solute concentration. Therefore, water in the ICF, okay, moves across the cell membrane, the semipermeable membrane, in the direction of the higher solute concentration. If the ECF solute concentration is high enough, this could cause a cell to shrink or shrivel. So again, the hypertonic solution is the ECF, meaning it has a higher solute concentration than is in the cell. Therefore, the cell loses water because the water moves across the cell membrane towards a higher solute concentration and will keep moving until equilibrium. But in this case, if you notice, instead of having that normal red blood cell shape, they're actually shrinking or shriveling. Okay, we don't want our cells in these two conditions. This one here, okay, falls under a category called water poisoning. And this one here falls under a category that we simply refer to as dehydration. Okay, so please remember, when you get into this topic on tonicity, we're still talking about the movement of water. That's why I put it by osmosis. It falls under osmosis, special type of diffusion. When you hear the term isotonic, hypotonic, or hypertonic solutions, we are referring to the ECF. Okay, these three terms are in reference to the ECF. And depending on the solute concentration of the ECF, depending on whether the ECF is behaving as a isotonic, hypotonic, or hypertonic, okay, that in turn will cause the net movement of water if the solute concentration between the ECF and ICF is drastic enough. So you could say we're talking about water balance of a cell. Now at this point, listen carefully. I've tried to put as many video links as I could on here. You need to watch these video links. It's hard enough to explain osmosis and tonicity and just diffusion in general in person. So I try to put as many video links. And not only did I do that, this time I put watch this video because I had a few students ask me, or actually I had a few students say, what? And I said, yes, there's video links. They're in bold type, they're underlined, they're usually colored. In a color, I think it's a blue font or blue colored font, okay? But on this chapter notes, I did remember to put down, watch this video. Please watch the videos, okay? All right, so we're still under membrane transport, but notice now we're in a type of membrane transport called carrier-mediated transport, okay? So what we're focusing on now are carrier proteins. Carrier proteins involved in passive processes of membrane transport, carrier proteins involved in passive processes of membrane transport. Okay? Now, when we go over this, if we are talking about carrier proteins involved in passive processes of membrane transport, you're going to come to know this as facilitated diffusion. Now, it's also showing you facilitated diffusion brackets on the channel, but we're not going to talk about that in reference to facilitated diffusion. I'm going to focus on just the carrier protein. Okay, so again, we're just focusing on the carrier protein. Thus, this type of membrane transport is called carrier mediated. It could be passive, in which case we call it facilitated diffusion, or it could be an active process, simply called active transport. In either case, they require a carrier protein. But keep in mind what I said. For passive processes of membrane transport, they may or may not require a transmembrane protein. If they do require one, it'll either be a channel protein or a carrier. Well, we're talking about carrier-mediated transport, so we're only focused on the carrier. If it's an active process of membrane transport, keep in mind what I said. It always requires ATP, because it always requires a transmembrane protein. Specifically, what we focus on again is a carrier protein. Okay, so for a passive process of membrane transport that always requires a carrier protein, facilitated diffusion. Again, they're showing you a channel because you can apply it, but for the test, I just want you to apply facilitated diffusion to carrier proteins. So just this example here. Okay, so for its facilitated diffusion, so here in the name of this type of carrier-mediated transport, okay, diffusion, that's letting you know it's a passive process. So not only is it not going to require ATP, it's going to require a concentration gradient. But we said this is a type of carrier-mediated transport, all right? Therefore, this is going to fall under a passive process of membrane transport. That does require a transmembrane protein, in this case, a carrier protein. The facilitated, in this phrase, term facilitated diffusion, the facilitated is in reference to the carrier protein. It's facilitating the movement of solutes from a high concentration across the cell membrane to a low concentration of solutes. So you can see this here. But it's called a carrier protein because it's physically going to be grabbing onto the solutes and moving them across. If you look over here for the channel, it just creates an opening, a pore, a doorway, to where once it opens, those solids that or ions that it's permeable to can pass through freely. Carrier protein's different. I am manually, physically going to grab a solid on one side of the membrane where there's a high concentration, and I'm going to move it to the opposite side or transport it to the opposite side of the cell membrane where there's a low concentration of solid. Well, if that doesn't make sense, I'll give you a specific example of facilitated diffusion. Okay. Carrier-mediated transport, facilitated diffusion. Okay, we say the facilitated here is in reference to the carrier here. Down here, what do we mean by facilitated and carrier? We're talking about a carrier protein. What's a specific example of a carrier protein? A carrier protein known as the glucose transporter. Okay. There are actually many different types of glucose transporters, but in general, you find glucose transporters in the cell membranes of our cells. Okay, and it's in the name. They're involved in membrane transport, but they just don't transport any substance. They only transport glucose. So after you eat a meal, or when you've eaten a meal, as what you've ingested, as those nutrients pass through your digestive system, roughly about the time those nutrients reach your stomach, okay, there are certain cells of your stomach that will signal your pancreas cells, certain cells of your pancreas, okay, to release the hormone insulin. Insulin will be secreted into your bloodstream. And insulin serves as a signaling molecule telling every other cell of your body, hey, the good stuff is coming, glucose. So you guys need to make sure you have not only these types of proteins inserted into your membrane, make sure you have enough of these transmembrane proteins called carrier proteins, or more specifically, glucose transporters in your membrane. Eventually, as your digestive system in your small intestine starts to absorb the glucose into your blood, from there, that glucose in your blood will then move out into various tissues, eventually reaching the extracellular fluids surrounding your cells. Now, you can see here we have a high concentration, okay, of the solid glucose compared to the inside of the cell where we have a low concentration of glucose. Okay, now the inside here, as we said, that's where we have our ICF. I didn't even draw the line that long, it just did it. Out here you can see we have our ECF, but they wrote it out. Now what happens is the molecules of glucose, since there's such a high concentration out here in the ECF, that increases the likelihood or the chance that a molecule of glucose will move into the carrier protein, which is open in the direction of the ECF. Now the carrier protein is going to have a receptor site, but I prefer to call it a binding site. Okay, the carrier protein is going to have a binding site, a shape to it that complements the shape of the molecule of glucose, so that the molecule of glucose now can act like a key, and this binding site in the glucose transporter acts as a lock. Once the molecule of glucose fits into that binding site, that triggers a change in the carrier protein's shape. Now the carrier protein goes from being open in the direction of the ECF to where it changes shape, and now it's open in the direction of the ICF. Now what you can't see is that when this carrier protein is changing shape, now opening in the direction of the ICF, this binding site loses its shape a little bit to allow for the release of glucose. Now this molecule of glucose is just diffused from an area of high concentration to an area of low concentration with the help of this carrier protein called the glucose transporter. And no energy was required. However, it is a type of diffusion. What was the driving force? We had an area of high concentration, and the sugar molecules are diffusing in a direction of low concentration. So in other words, we have a concentration gradient. Okay, that's facilitated diffusion, a type of carrier-mediated transport. But then we have active transport. Okay, active transport is going to involve a carrier protein. The catch is this, everybody, something I forgot to mention. Okay, well, I didn't make it stand out. Passive process of membrane transport. I said, what is the driving force? We have to have a concentration gradient. For active processes of membrane transport, I never said that. Well, that's because for active processes of membrane transport, we do not need a concentration gradient. As a matter of fact, Active processes of membrane transport move substances against the concentration gradient. So something I forgot to mention, you can think of passive processes of membrane transport as if you go to the Guadalupe and you get on the inner tube and you go with the current. You just passively float down the river. You let the river do all the work. You don't have to use any energy. But you can think of active processes of membrane transport as in you might have done this. You finally get to the end of the line. You get out of the river, but it felt so good and you feel there's time. You get the bright idea of let's go back up to the beginning and let's do this again. And then you realize what? This is taking a lot of energy. I'm having to actively move, okay, against the current if you actually move through the water. So that's the way it works. So for carrier mediated transport, okay, the type of active process that is a form of carrier mediated transport is simply called active transport. Okay, active transport requires a carrier protein that will move solutes against the concentration gradient. Therefore, since it's moving against the flow, it's going to take energy. So notice on this figure, it's showing you this carrier protein is moving a solute from an area of low concentration in the direction of an area of high concentration. It's going against the concentration gradient. Well, this is because our cells, okay, it's not just an issue of high versus low concentration. For certain solutes, our cells say, look, I have a low concentration of this substance, but it's not low enough. I need it lower so I can do my job. And then sometimes our cells will say, you know what, in my ICF, I have a high concentration of this substrate, but it needs to be higher in order for me to do my job. So this is why I chose this example. An example, okay, of active transport, okay, which is a type of carrier mediated transport, all right, is this carrier protein. There is a carrier protein who has a long name. This carrier protein's name is a sodium/potassium ion exchange pump. If you take AMP, you should absolutely cover this, at least with the nervous system is where it's most popularly covered. So here it is. So now we're talking about a type of carrier-mediated transport that is called active transport. It involves a carrier protein that is actively going to move sodium ions and potassium ions across the cell membrane, except when it moves the sodium ions, okay, it's moving the sodium ions from the ICF into the ECF, where there is already a high concentration of sodium. And it is moving potassium ions from the ECF to the ICF, where there is already a high concentration of potassium ions in the ICF. Okay. Since this transmembrane protein, more specifically a carrier protein called the sodium-potassium ion exchange pump, since this protein is moving ions, okay, in the direction of an already high concentration of each of these solutes, it's going to need energy. So the way it works is for this carrier protein called the sodium-potassium ion exchange pump, for every 1 molecule of ATP, it will pump out 3 sodium ions into the ECF where there's already a high sodium concentration. And like a revolving door, it'll grab 2 potassium ions from the ECF and pump them into the ICF where there is already a high potassium concentration. You see this a lot. You hear talk about this primarily for your nervous system. Your nervous system is made up of 2 general types of cells: glial cells, supportive cells, very important, and your neurons. Your neurons are the ones that do the talking, the communicating, the regulating. And it's your neurons, okay, that need these pumps to be working, because your neurons cannot communicate and regulate body functions and talk to other cells unless the potassium levels inside of them are not only high, but high to the point that they need it. And in addition to that, those neurons not only need sodium levels to be low in the ICF, they need them to be really low, especially in comparison to the ECF, so that these neurons can perform their functions, okay? I'm not going to go into more detail than that. They will in A&P, but just keep in mind this one here, okay? Now, our last type of membrane transport is called vesicular transport, okay? If you look here, vesicular transport involves the movement of substances contained within vesicles into and out of a cell. These vesicles are derived either from the cell membrane or from the Golgi apparatus. Now listen carefully. The compounds we talked about that form your cell membrane, those are the same compounds that form the membrane of your membranous organelles, such as the nucleus, the rough ER, and the SER. Those are also the same compounds that form the membrane of vesicles. So I'm going to use this figure here. This is the simplest representation. This is the simplest representation of a vesicle. Let me see if I can spell this, okay? Simplest representation. I often refer to a vesicle as a tiny lipid package. Mainly because if you recall, I just said that the compounds that form the membrane of your cell membrane are the same compounds that form the membrane of your membranous organelles, and those are the same compounds that form the membrane of vesicles. So I describe vesicles as a lipid package because the main compound that forms your cell membrane, and thus the membrane of the vesicle, are your phospholipids. But I call it a lipid package because these phospholipids, as well as the other compounds, proteins, which you'll find forming the vesicle, okay, are basically enclosing a little package, a package of solids. All right, now, vesicular transport. This type of membrane transport is required whenever the solute is too large for any channel or carrier protein to transport across the membrane. Or this type of transport, vesicular transport, is required when a cell needs to transport a large quantity of primarily proteins across the cell membrane. Okay, so again, it's either the size of the solid is too large for any channel or carrier protein to accommodate, or it's the quantity of solids. Okay, the quantity is too great for any channel or carrier protein to accommodate. Therefore, we need this type of transport. There are three types of vesicular, or there's two types of vesicular transport: endocytosis, endo meaning within, and exocytosis. Okay, the exo meaning out or exiting out or outside. For endocytosis, there are three forms of this type of vesicular transport. There's a type of endocytosis called phagocytosis, another type called pinocytosis, another type called receptor-mediated endocytosis. For receptor-mediated endocytosis, first of all, this is a selective type of endocytosis, a selective type of vesicular transport, okay? Receptor-mediated endocytosis, the receptor is in reference to transmembrane proteins that function as receptor proteins, okay? These receptor proteins are required for this type of endocytosis, this type of vesicular transport. And what it is, I'm going to use an example to explain it. I think mostly everybody's heard of particles called LDL. There's LDL and HDL. HDL is the good form, high density lipoprotein. LDL is the bad form, or the worst of the two forms, or the bad form, I'm sorry, just the bad form, called low density lipoprotein. LDL is actually more specifically called a particle. It's a large particle. It has a phospholipid coat with its own proteins embedded in it, but this phospholipid coat and proteins, okay, form an outer layer that's encasing or enclosing a high concentration or a high number of molecules of cholesterol, okay? Cholesterol itself is a pretty large molecule, but when you're talking a large quantity of them, that makes it even harder for a cell to move across. Our liver cells process these particles, okay? So what happens, in order for our liver cells to transport these LDL particles across the liver cell membrane, since they're so large, the liver cell has to transport these LDL particles through receptor-mediated endocytosis. The liver cells will have receptor proteins, transmembrane proteins, that have a shape facing the ECF that complements the shape of the proteins in the LDL particle. Therefore, these proteins in the membrane of the LDL particle, okay, will bind to the receptors. And when enough receptors in the liver cell membrane bind to enough of these proteins on a single LDL particle, this will trigger the cell membrane of the liver cell to begin to, what they say, invaginate or fold inward. As the membrane folds inward, as the arrows are indicating, okay, what you can't see clearly is that the membrane to the edges is being pulled over the top as this is being pulled inward, or drawn inward, I should say. Okay, that way the membranes on each side over here will eventually come together, which they're not showing you, but over here they're trying to show you. They will eventually come together so we don't end up compromising the cell membrane and thus killing the cell. The cell membrane will overlap, therefore it stays enclosed as a piece of the membrane pinches off, becoming the membrane of the vesicle. Inside we have our LDL particle, and now the liver cell can process it. This is why I say, okay, you can see here to explain further what I mean by the compounds that form the membrane of our cells. These are the compounds that form the membrane of our vesicles, except in the case of receptor mediated, you are definitely going to have specific proteins which have attached to large particles in the ECF, helping transport this large particle across the membrane into the ICF. Now, a second type of endocytosis is called pinocytosis. This is non-specific. I don't even have a clear exact example of any cells of our body, but it is known. Cells of our bodies, for some reasons, okay, well, sometimes the cell membrane of some of our cells will begin to fold inward, drawing in solids that are next to the cell or neighboring solids. As the membrane pinches in, the outer edges of the membrane start to overlap to where these two ends of the membrane will fuse together so we don't have a break, which is here. They just forgot to show another arrow over here where the membrane fuses. Okay, so what's here and here, what's here and here is now this here, and then this piece of the membrane pinches off. And what will happen is our cells will just draw in a large concentration of solid and then sort through it to see, hey, is there anything I can use? And anything it can't, it'll pass it on to a lysosome to break down what it doesn't need. So we call this pinocytosis. Pinocytosis is non or not selective. Okay, it's just the cell sampling what's in the neighboring ECF. The third type of endocytosis is called phagocytosis. Phagocytosis is selective. Okay, phagocytosis is most commonly performed in general by our white blood cells. We have white blood cells such as macrophage monocytes. Macrophages, white blood cells in your tissues, monocytes, white blood cells in your blood, and they are capable of phagocytosis. The lay term for phagocytosis is eating something up, but I want you to know phagocytosis. The reason why I say this is selective is because white blood cells are scanning either your tissues or what's in your blood for anything they deem foreign or a foreign object. And if that white blood cell detects something that it considers foreign, it will endocytose that foreign object. Okay, say this is a bacterial cell such as an E. coli, which has a bacillus shape. It'll endocytose that foreign object, encasing it within a vesicle, except in phagocytosis, whatever the cell phagocytizes, okay, whatever foreign object the cell phagocytizes, that substance will always be taken to lysosomes. If you recall, lysosomes have degradative enzymes or digestive enzymes called acid hydrolases, and their job is just to tear up and destroy whatever they come in contact, but they work great at a pH of 5. However, these things are like attack dogs, these enzymes. Therefore, a cell keeps these enzymes encased in an organelle called the lysosome. But for phagocytosis, white blood cells in general perform this type of endocytosis. It's selective because only thing being endocytosed is whatever is considered foreign to your body, and that foreign object, once it's endocytosed into a vesicle or phagocytized into a vesicle, it will be transported to lysosomes to be destroyed. Sorry, I'm getting tongue-tied, everybody. It's late for me now. I'm getting old, God. Okay. So vesicular transport, there's two types. Endocytosis, of which there are 3 types. And then the second type of vesicular transport is called exocytosis. So when you go over vesicular transport first break it up into two types, endocytosis and exocytosis. And then under endocytosis, divide endocytosis up into the three types I just talked about, okay? Kind of create an outline for yourself if it helps. For exocytosis, there's no three types. There's just one type, so that's the good news, okay? But listen, when we go over exocytosis, if you did your studying of the organelles, this should start to sound familiar, okay? If you recall, when we went over the membranous organelles, we talked about the rough ER, which they're showing you here. It says ER, but this is a rough ER. The rough ER is where you find ribosomes attached to a membranous organelle called the endoplasmic reticulum. And the ribosomes will synthesize proteins, and the ER will modify those proteins. One type of modification is adding sugars. Then what will happen, okay, is a piece of this, what we call rough ER, the rough because of the ribosomes attached, but a piece of the rough ER's membrane will pinch off, forming a vesicle actually, and it'll be transported to another organelle called the Golgi apparatus. Okay, so we have specifically the rough ER or RER. Oh, that just sucked. Sorry. So we have our ER, but specifically this is the rough ER. Sorry, everybody, if you're sensitive to S words. Gosh. Okay, I'm trying not to curse. So we have the RER here, and then over here we have the Golgi apparatus. And I went over this in the notes in chapter 6. Any protein synthesized and modified by the rough ER will be transported to the Golgi apparatus, where those proteins will be further modified. Then the Golgi apparatus' second function, besides modification, its second function is to sort and transport proteins. And I put on there, I told y'all, that when the Golgi apparatus sorts these proteins that it further modified, it either sorts them and packages them into organelles called lysosomes, or it transports these proteins to the cell membrane. When it transports them to the cell membrane, the second place it'll sort them is by inserting them into the membrane. However, we are talking, I'm sorry, that's not even the right one. However, we are talking, here's a protein here. So this one has been sorted, becoming a transmembrane protein. However, I said that the third direction, okay, that the Golgi apparatus would sort proteins is by releasing or secreting them into the ECF. Listen carefully. Exocytosis is in reference to this third one here. Exocytosis has to do with the transport of proteins from the Golgi apparatus in a vesicle to the cell membrane, and then those proteins are released, exocytized, or secreted into the ECF. So exocytosis is only in reference to those proteins that they've labeled secreted proteins. Okay? I'll be honest with you, in advanced cell molecular classes, it's rare you hear exocytosis. They'll just say protein secretion or the protein secreted proteins, okay? But again, exocytosis begins at the Golgi apparatus. Proteins modified by the Golgi are then sorted and packaged in the vesicles, transported to the membrane, and released or secreted into the ECF. That is exocytosis, just where they're released into the ECF. So that's what this figure's up here, just to show you these, okay? I know this is a tough chapter. I wanted to get it all done as one and not break it up, okay? So please make sure you watch the video on this, the recording. Please watch the video links, at least by this weekend. Watch them to help you out, especially if you're going into the allied health field, especially for diffusion and osmosis, okay? All right, everybody, y'all take care, and I'll talk to y'all later. Bye.